Hello, class. This is a recording for Biology 105, recorded on August 29th, 2025. I'm unable to be there in person today, but we're going to just resume our lecture material so that we don't fall behind anywhere. But here we are in the week one, right? I hope your labs all went well. Don't forget that you do have a reading quiz due this week. That reading quiz is 2 attempts, 60 minutes, open book, open note. It's just meant to make sure you're engaging with the material. The answers can be found online, in the textbook, or in the PowerPoint. All right, and we are going to now get back into the actual presentation. So last time we finished up talking about our introductions and cell types and things like that, or prokaryotic versus eukaryotic cells, the domains of life, all these things, okay? And we're going to now get into some discussion of the scientific method, basic statistics talk, just to, I'm not going to really test you on that, but I think it's important information. What a primary research article is, just the foundational concepts. We're going to knock as much of that out today as we can. All right, checking my start time. There we go. Okay, we'll go about 40 to 50 minutes on this lecture. So in this PowerPoint, we're going to be talking about the scientific method, how that works, what's a hypothesis, what's a theory, how do we design an experiment, what's a primary research article look like, and just very, very basic statistics. I'm going to mention it again. I'm not going to be testing you on the statistics piece. I just think it's very important that you get it regularly, you get these explanations regularly in a lot of different courses. Okay, once again, starting a presentation with a definition. It's old technique, it's sloppy, but we're doing it anyways. The word science is derived from Latin and means to know. So the scientific method is a way of answering questions. It's a way of explaining. It's a way of understanding. The scientific method takes an observation, proposes an answer, and then analyzes that answer. And if the answer holds up to analysis over and over and over again, eventually it becomes a theory. So the scientific method is not the only way of knowing, but it is the way of knowing that we use in the sciences. It involves observation and then experimentation. Science in general has this loop here at its center, okay, of forming and testing hypotheses. You make observations and form hypotheses, which are explanations for those observations, and then you test them, then you form new ones, and you keep going back and forth. And ideally, science is connected to the broader community in ways that are it's not a one-way street either. There are societal benefits and outcomes of scientific findings. Exploration and discovery can be encouraged by scientific findings, like in community science or something like that. Also, exploration and discovery carried out by non-scientists can be used to inform hypotheses, and drive the generation of new hypotheses. All right, this can give us some observations to ask why about. Then there should be some integration with the community. Okay, the community analysis and feedback about what do we think about these findings? Can we incorporate these findings into how the community operates? What other questions do we have? All right. So what is a hypothesis? Textbook definition right here. A tentative answer to a well-framed question. Okay. Now a good hypothesis, okay, needs to be testable. So a scientific hypothesis needs to generate predictions, okay, that you can then test with an experiment or with observational experiments. Okay? So, a good hypothesis, not only does it need to be testable, it also needs to be falsifiable. So, if I propose an answer to a question that's reasonable and well framed, there needs to be a way to prove me wrong. Otherwise, my explanation isn't really acceptable, right? The burden of proof always lies on the person making the claim. So we'll talk about a fun example of hypothesis testing in a future slide. But oftentimes there will be multiple hypotheses, multiple testable answers, okay? Multiple answers to the well-framed question. We'll use an example of fly feeding patterns and fly life cycles that should be useful. And one of the big ones here is that failure to falsify a hypothesis does not prove that hypothesis. The scientific method is set up to where you are supposed to design experiments that could disprove your hypothesis. So you put forth an answer, and then you design experiments that try to prove you wrong. And if those experiments fail to prove you wrong, well, then that provides support for your hypothesis, but it does not necessarily prove your hypothesis. You need additional rounds of experimentation. But if a hypothesis has been tested and tested and tested and tested, and no one's been able to prove it wrong, then it can grow up and become a theory, eventually becoming a law, which is a theory that's even more accepted, but we don't have to get there just yet. So this is an image from your textbook talking about how you test ideas, test hypotheses. It's really just about making a good hypothesis, having predictable results, conducting experiments, gathering data, and analyzing the data. But then there's this interpretation piece. So the results, the data that you collect, it might support your hypothesis, contradict your hypothesis, or inspire new hypotheses. And that just sort of goes around in a loop over and over again. I like this image for the scientific method, but you'll see hundreds of images similar to it if you do a Google search for scientific method. All right? So you start with an observation. You see something in the world around you, you see something in the lab, and you want to understand why it happens that way. All right? You construct a hypothesis, an answer to that question. You test it, you interpret your results to draw a conclusion, and you either accept or you fail to reject, or you reject your hypothesis. All right? If your hypothesis is rejected, construct a new one and try it again. Eventually reporting results, hopefully reporting results whether you accept or reject your hypothesis. Okay? And just because you accept your hypothesis here doesn't mean that it's necessarily true. You just didn't disprove it in your experimentation. So this is a pretty standard scientific method in action explanation here. Let's go through the steps with fruit flies. So here's Drosophila melanogaster. All right, Drosophila is what we call a model organism. It's something that has been used in a lot of experimental studies. A good model organism is very well understood. In plants, the classic model organisms are Arabidopsis thaliana, as well as tobacco. A specific species is usually used. Now, all of these species share a few things in common. The biggest one I've already mentioned. They've been used in many studies, so it makes it easier to reproduce results between the studies. You're trying to control for a lot of variables here. So you do the same study in the same organism, it's more likely that your results will be comparable. Okay? You want to be comparing flies to flies, right? Not flies to oranges. The other benefit of at least these three species is that they have been sequenced. Their genomes have been sequenced, so we understand what their genetic code at least looks like. We may not know what every portion of the genetic code encodes, but you know what the genetic sequence is, and for a lot of reasons that we'll discuss probably more in lab, that's very beneficial. Okay, so you've got a well-studied organism that has been sequenced, and a good model organism also has a bunch of specific lines generated. So there are different varieties of the model organism, say ones that have certain genes removed, or mutants that have, say, no wings for a fly, or different colored eyes for a fly, or a different life cycle length or behavior. Now, all these different mutant strains that have been generated, those allow for more nuanced research work. This will come up in other classes that you take for sure. But always feel free to reach out and ask me if you want to know more about these different mutants and their use. So here we go. We've got Drosophila melanogaster. You make an observation that the life cycle of one of these flies is about 30 days at 29 degrees Celsius. You develop a question. You have some preconceived information. Okay, not preconceived, but you have knowledge going into this. You know that temperature affects the rate of chemical reactions. Higher temperatures represent more kinetic movement of molecules, so it tends to make reactions happen faster. So you think about this and you say, well, how would a change in temperature affect the life cycle, the length of the life cycle of a fruit fly? So you make an observation and you develop a question. The question being, how do changes in temperature affect the life cycle of a fruit fly? Or do changes in temperature affect the life cycle of a fruit fly? Next, you propose a hypothesis. This hypothesis here is that decreasing the temperature of a fruit fly's environment will increase the time it takes the fruit fly to complete its life cycle. You propose this hypothesis based on pre-existing knowledge, right? That gave you the directionality of the hypothesis. Instead of saying that it would change it, you're proposing specifically that decreasing the temperature will increase the time it takes the fruit fly to complete its life cycle. If you had multiple alternate hypotheses, one could be that decreasing the temperature will increase the time it takes, and the other could be that decreasing the temperature will decrease the time it takes. Those would be two alternate hypotheses, okay? There's also something important here. It's not mentioned in the slide, but I'm going to mention it right now, and that's the null hypothesis. And the null hypothesis in this case would be that temperature will not affect the life cycle of the fruit fly. So the null hypothesis is that there is no effect of the thing that you are manipulating on the thing that you are measuring. You as the researcher are manipulating the temperature and you are measuring the length of the life cycle of the fruit fly. There's your hypothesis here that you've generated. Colder takes longer to finish its life cycle. Next step here, you do an experiment. 100 fruit flies are placed in a cold treatment. 100 fruit flies are placed at a 29 degrees Celsius or normal treatment, the one that you observed in the natural system. So if the hypothesis is correct, all right, which, we want to be careful with that, but the fruit flies at lower temperature will complete their life cycle after the ones at the higher temperature. That would be evidence to support our hypothesis. It would indicate that temperature has some impact on life cycle length in fruit flies. Step 4, you analyze the results. You find, great, the fruit flies placed at the lower temperature took longer to develop. You publish the results, or you just put the process through peer review, and you find, there's some alternate explanations here. So one alternate explanation could be that when it's colder, it's harder for the fruit flies to digest their food. It's not as soft, makes it harder for them to eat. So you have to do an additional experiment with more control, where you have to provide a food supply that doesn't change consistency as you change temperature. All right, so now you go back, you retest the hypothesis with a different experiment. You see if it's the same. You see if you see if your results continue to support your hypothesis, or if you're not able to disprove your hypothesis once again. Statisticians, people who are real sticklers for experimental design, they'll be sticklers for that sort of language, and it is important. Just providing support for your hypothesis doesn't prove it. All right? Solid. Designing a good experiment. Okay? I'm going to talk to you about these in some other slides that I've got for you as well. But we'll introduce them right here, and then I will come back and repeat them again. They're just that important. So, a well-designed experiment. What does it have? And I want to also preface this with, if you're doing experimental design, you should be able to identify these in your experiment. It's easy to propose an experiment and say, I'm gonna take a bunch of sunflower seeds and water them with a solution of water, and then a solution of Gatorade, and then a solution of vodka. And I'm gonna see how that affects the growth rate of the sunflowers. Okay? But you need to be able to ID what's my independent variable, dependent variable, experimental groups, what do I have a control group, what is my control group, all of these other things need to be understood when you're designing your experiment. So in this case, the solution that I'm irrigating the sunflowers with is my independent variable. It is the thing that I, as the researcher, am manipulating. Or in other words, I'm saying that the growth rate of sunflowers depends on what they are irrigated with. So the dependent variable depends on changes in the independent variable. A variable in general is just something you can measure. I can measure or identify what my solutions are, right? And in an experiment like this, you probably do a water group and then a 10% Gatorade, a 50% Gatorade, 100% Gatorade, 10% vodka, 50%, 100%, right? So you have a bunch of treatments, all right? The independent variable would have a lot of different values. Water, 10% Gatorade, 50% Gatorade, 100% Gatorade, a bunch of little categories. The dependent variable you'd measure as height of plant after 15 days or increase in plant height over 2 weeks. There's a lot of different ways to represent this, something you should be thinking about. But you're proposing that the growth rate of the sunflower depends on the solution that you are irrigating with. Hopefully that makes sense for dependent versus independent variables. Now, oftentimes in a study, you're going to have experimental and control groups. And that can be tricky, okay? Because not every study has a control group. But the control group is the group exposed to regular conditions. I want to see what sunflower growth looks like normally, okay? So in my example here, irrigating the sunflower with water is sort of the control group, where normally the sunflower would grow at this rate. Everything's controlled. That's the control group. It's what we're comparing. Our other groups are experimental groups too. So the experimental groups are those that receive the experimental treatment. All right. The control group is the group that receives the normal the normal condition, the normal set of independent variables. All right. So in this experiment, once again, we're proposing that irrigation with these solutions is going to affect sunflower growth. But how do we know if it's affecting sunflower growth? Well, we need to know what the normal rate of sunflower growth is, and that's why a control group is useful. Positive versus negative controls are complicated, and I'm going to bring them up in a slide that you're about to see. Experiments need repetition, okay? You saw in the fly experiment, we used 100 flies instead of just using one fly that was kept cold and one fly that was kept at warm temperatures. That's because there's natural noise, okay? There's chance, there's random things that could happen inside the fly, and there's natural variability that could change how long its life cycle takes. But taking averages and using replicates, okay, using replication is really key. So a good experiment needs replication. That number of replicates can change depending on what you're studying, you know? In plant ecology or in plant phys, yeah, in turf grass science, at least, I can tell you from my own experience, it's usually sufficient to have 4 replicates of each treatment, but 6 is done regularly as well. There are reasons that having 4 replicates is a good default number. It has to do with just how the math works for generating the statistics. So in your head, it's good to think 4. Because really, you must have 3 replicates for any of the math to work, and sometimes one of your replicates dies or has issues. You also need the study to have repetition because in time and or in space and slash or in space. where the study needs to be repeated, the whole study. Because if you get results once, you want to show that you can get the same results again. Experiment needs to be well defined. You need to very clearly lay out your hypothesis and what your independent variables are, and why you think the independent variable will affect the dependent variable, and how you will know if you are supporting or rejecting your hypothesis. You really got to know what the results that you expect are. You need a well-defined experiment. It's a very broad statement. It's kind of pretty obvious. Two big types of data. All right. Data falls into two categories. You've got qualitative data, you've got quantitative data. All right? Qualitative data is a description of something. So we've got here Jane Goodall with observations of chimpanzee behavior. Okay? Now, one way to think about this is two individuals collecting qualitative data may come to completely different conclusions. Two individuals making different observations of chimpanzee behavior may say that a chimpanzee was happy or sad or angry or scared. Two different people, same chimpanzee, same behavior, different interpretations. So qualitative data is difficult, okay? Qualitative data can include, I think that the chimpanzee behavior example is very good. We'll just stick with it. It's fun to think about sometimes, like, oh, is that qualitative or quantitative? But quantitative data is specifically data that can be measured. It's got a number associated with it. And quantitative data is useful because it can be analyzed statistically and mathematically. There are ways to statistically analyze qualitative data, but oftentimes you convert qualitative data into quantitative data. Like, for example, in a focus group. In a focus group of people, you take 50 people, you give them a candy bar, you ask them how they felt after the candy bar. Or you say, how based on this statement, how much do you agree on a scale of 1 to 5? This eating this candy bar made me feel full. Rate your response on a scale of 1 to 5. 1 being fully disagree, 5 being fully agree. All right? Now, what you've done is you've taken a qualitative data sort of response, but you've recorded it as quantitative data. And now it can be analyzed using classic statistics. Okay? So quantitative data is something you can measure. And repeatable quantitative data, or like real, I'm going to go out on a limb and say like real quantitative data that when you haven't sort of tricked everything into looking like quantitative data, is something that any researcher, if they're measuring correctly, they'll measure it the same way. So plant height, right? As long as you've got a clear protocol for where you start measuring plant height and how you measure plant height, two people measuring plant height will measure the same height of the plant. You see what I mean? That's truly quantitative. Nice number. Everybody could collect it the same. It's great because you don't need, you can have a team of researchers working together. You don't have to have the same person measuring everything. Okay, great. Fantastic. Sometimes the challenge in doing good experiments is to collect both qualitative and quantitative data. There's so much to think about. So much to think about. I'll do a quick example. Quick example from my own research. In my research, you need to evaluate turf grass quality because you want to see if you can, in my work mainly, you put some biochar in the soil with the hopes that you can keep the turf grass looking nice with reduced irrigation. The hypothesis being that increasing concentrations of biochar in the soil will allow us to irrigate less while still maintaining an acceptable aesthetic quality. That's sort of my proposal here, my hypothesis, right? Now, to test that, you need to be able to measure turf grass aesthetic quality. And there's something called the NTEP, or the National Turf Evaluation Program, which trains you to measure the quality of turf grass visually. It's on, I believe, a 1 to 9 scale or 1 to 7 scale. It's been a long time since I've had to do it. But you look at a patch of grass and you say, that's a 7. Ooh, that's a 4. Okay? There's also equipment, quantitative equipment, that can measure how green something is, the turf grass is. Or you can directly measure the photosynthetic health of turf grass using handheld equipment, reflectometry equipment, and stuff like that. I'd love to talk to you more about it, of course, if you're curious. This is my 6 years of my life. The point of the matter is, one's qualitative, but it's numerical, so now it's quantitative. The tricky one, right? One, and then the other two, the other measurements are purely quantitative. Something to think about. Something to think about. So, negative versus positive controls. All right. Negative controls are the ones that you're probably most familiar with. In a negative control, an example would be like the plant growth rate with the different amounts of Gatorade and vodka, right? I'm saying that Gatorade and vodka are going to reduce plant growth rate realistically, okay? But we need to compare to something else. So a negative control keeps everything except the treatment. All right, it's identical to the experimental treatments, the experimental groups, except that you omit the thing that you think has an impact. This controls for chance and just random variability in sunflower height, for example, here. And negative controls are essential. They're the ones that we see the most. Cool. Now, this includes things like a placebo group in a pharmacological study. All right, you're trying to see if this pill makes people's headaches go away, so you give some people a sugar pill, they take it, it should have no impact, and that way you're controlling for natural cessation of headache symptoms or the effect of just taking a pill and thinking that it's going to help you. Okay, so negative controls, they don't receive the experimental thing. So in this experiment, thing A causes B to happen. So you exclude A and you make sure that B didn't happen for another reason. A positive control is different, okay, in that you're usually experimentally excluding something. So let's say that you are, yeah, let's say that you are, I want to get a nice image here. Perfect. Let's say that you're testing if some different compounds can inhibit bacterial growth. So you take a nutrient agar plate, something that you've seen in lab now, with a bunch of bacteria in it. All right. You take all these discs that have potential antimicrobial agents, and you're testing, hey, do these all work to inhibit the growth of this bacteria? Then you just put it in the incubator and you let it sit, and you see, like, okay, did the bacteria grow? And if the bacteria didn't grow around the disc, you propose that the bacteria were killed or their growth was inhibited by the antimicrobial agent. Pretty straightforward, classic bacteriology work. Now you want to also have controls here. It's possible that just having the paper disc with some liquid on it inhibited bacterial growth. So you want to probably have a disc, for example, that's just got water on it, the same volume as the antimicrobial agent that was placed on each one of the discs, but just distilled water, so that you can see if the paper disc itself was actually inhibiting. Let's take a look here. So in this example, we've got disc A, B, and C. They've got different compounds on them. Disc A is a negative control, okay? That means this is just water, probably, all right? Disc B has the thing that you are testing. But in this case, we've got a positive control incorporated. That's disc C. Disc C has a known effective antimicrobial compound. So we can say, okay, we've proven that on this disc, all right, just a disc of paper, a piece of paper with water on this Petri plate won't inhibit the growth. That's good. That's the effect of our negative control. Our positive control has shown that, okay, these bacteria are sensitive to antimicrobial compounds. and this disk application method works. And then disk B here shows that there was some effect of the compound of interest. So positive and negative controls were both used in that experiment. You need both of them. So you need to ensure that A can cause B. So you use a positive control. Right? They're also used in genetic experiments in a lot of different ways that are very, very interesting. More and more genetic experiments involve silencing genes to determine what they do. Okay? And that involves having negative controls and positive controls. It's all fascinating. Come talk to me if you're curious about it. I'd love to talk more about it, but we need to continue moving. Let's check our recording time right now. So we have hit 30 minutes. We'll do about another 10 to 15, and we'll call it a day. So here's what these little plate with this method actually looks like. It's awesome. Great. So in this experiment, we need to know what the independent, the dependent, the control group, all this stuff. What is your null hypothesis? Let's talk about it. So the independent variable here is the different antimicrobial compound. All right. The dependent variable is meant to represent how good the microbial compound is at inhibiting the bacterial growth, but it needs to be something quantifiable. So in this sort of experiment, what you'll do is oftentimes measure the actual zone of inhibition. Okay, so the size of the zone of inhibition could be measured as a quantifiable variable of how good was the antimicrobial compound at inhibiting growth. Okay, so what are you changing? What is the researcher manipulating? It's the presence of the antimicrobial compound. What are you measuring to see if it had an effect? It's the size of the zone of inhibition. Or you could just present it as presence or absence of the zone of inhibition. Was there a zone of inhibition? If there was, give it a value of 1. If there was not, give it a value of 0. This gives you binary data, which can also be analyzed and is also quantifiable. Okay? Or quantitative, my apologies. If you had a bunch of replicates of this experiment, you would say which percentage of disks resulted in a zone of inhibition, possibly. There's a lot of ways you could measure this, but the dependent variable is the zone of inhibition. I hope that makes sense to people. Our control group here, all right, our negative control was the disk that just had water on it. It shows that the disk itself isn't causing any harm to the bacteria. The positive control in our example was the disk that had a known effective antimicrobial compound. You want to know that this technique worked and that, anyways. If for example, the known antimicrobial compound did not produce a zone of inhibition, you would know that something went wrong in your experiment. And then the fact that the antimicrobial compound you're testing didn't create a zone of inhibition would not confuse you or make you assume that it had no effect erroneously. The null hypothesis, what is it? Remember, our null hypothesis is, okay, and I'm not positive I mentioned this already. I'm just going to say this right now. Your null hypothesis is always going to be the same thing. It is always that the independent variable has no effect on the dependent variable. So the independent variable being antimicrobial compound, the dependent variable being size of zone of inhibition or presence of zone of inhibition. Okay? So your null hypothesis would be microbial compounds have no effect on the zone of inhibition size. You can phrase this very differently and say that the antimicrobial compound will not inhibit the growth of the bacteria. Okay? But your null hypothesis always boils down to the independent variable has no effect on the dependent variable. So if you're good at identifying your independent and dependent variables, you never have to worry about writing a good null hypothesis because it's just independent variable has no effect on dependent variable. I would normally clarify that final point on the board, but I've got no board here. It's all just on a screen. Hypotheses and scientific theories. All right. So I've got some additional slides. These are new for this semester that I've popped in here about statistics and hypotheses. I hope you find them interesting. I wanted to codify them. This is stuff that I always talk about. So, a hypothesis. What is a hypothesis? I believe we've already sort of talked about it as a testable explanation. Okay, null hypothesis, what is it? Independent variable, there's no impact on the dependent variable. I already said that part. Okay, there's a format for hypotheses that's really useful. It was taught to me back when I was a student taking bacteriology at then Humboldt State University. And it's the if blank and blank then blank format for hypothesis generation. You will need to generate hypotheses at some point in your scientific career, even if it's just for classes. And doing it this way, or forcing yourself to use this framework, will make sure that you're generating good, testable hypotheses and that you know the expected results that will support your hypothesis. So let's think back to the fly experiment. If blank, if temperature affects the length of life cycle for fruit flies. That's your hypothesis, right? Your alternate hypothesis that temperature affects life cycle of fruit flies. And I expose one group of flies to cold 18 degrees Celsius temperature and another group of flies to warm 29 degrees Celsius temperature. There's your experiment. Then I expect that the group of flies exposed to 18 degrees Celsius temperature will take longer to complete their life cycle. Those are the expected results if your experiment's true. It's really hard to convince students and convince anybody to do this because it feels like more work, and it is more work, and it doesn't make a nice, pretty, cohesive sentence. You can rewrite it when you submit it for an assignment, but doing it in the if blank and blank then blank format will make sure that you understand your hypothesis, and I really encourage you to try it. So I've already talked about null hypothesis. A theory is just a hypothesis that has been tested a bunch, hasn't been disproven, and people agree on it. They consider it to be reliable. So here we've got the standard issue. It's just a theory, disclaimer that a theory is an important word in science. Okay, theory means a lot. So right here we've got in 2016 a reporter stated that researchers were testing the theory that Zika virus caused birth defects. Why is that inaccurate? Well, researchers were testing the hypothesis, and after repeated rounds of testing, that hypothesis could become a theory. But they have a hypothesis. Theory just sounds more official, but using theory for things that aren't theory weakens the phrase theory. So scientific theory is a lot more important than it means a lot more than it's just a theory. Cool. Okay, statistics. You will have one lab that deals with statistics near the end of this semester. You're going to be doing a chi-square analysis. It's going to be dealing with Hardy-Weinberg equilibrium. I love this lab. It's the only statistics that you'll encounter in the course. So I'm going to mention statistics once here, and then it will come back again at the end of the semester. All right? But statistics does not need to be that complicated. Okay? Statistics does not need to be that intimidating. If there's one thing you might know from statistics, it's probably the p-value. And you probably, usually when I talk to students, if we were in a classroom, I'd say, what do you know about the p-value? And usually people say things like, if p is less than 0.05, that's good. You know, sometimes people will say, oh, we accept our hypothesis if p is less than 0.05. It's all fine. It's all fine. But I'm going to actually define p-value so you know what it means, okay? And you'll never have to be confused again about the output of a statistical test. Because most statistical tests, what they do is they give you a p-value, You need to be able to interpret that and understand what it means. Okay. So most statistical tests, if you check out this first bullet point, they calculate the probability that your observed results could have occurred by chance. In other words, they tell you what is the probability of receiving the results we did in our experiment if the null hypothesis is true. I'm going to repeat that. What is the probability of receiving the results we did in our experiment if the null hypothesis is true? In order to support your alternate hypothesis, you want this number to be very low. You want it to be unlikely that the null hypothesis is true. All right? So that's why a p-value needs to be less than 0.05. A probability of 0.05 is a 5% chance. So a p less than 0.05, one last time, indicates there is less than a 5% chance that we would have received the results we did in our experiment if our null hypothesis is true. And if our null hypothesis is true, our hypothesis is not true, right? Sample size is really important. Large sample sizes are better than small sample sizes, but don't let this turn you into a researcher who always does 20 replicates in every study that you do. Excessive replication is its own issue. So, statistics. Let me check our time one more time. I know I did that not long ago, but I just want to make sure we're at 41. Oh, that was not long at all. Okay. Statistics. It's difficult, okay? When you take your biostats class at Cal Poly Humboldt. Whatever statistics course you took, probably spent a lot of time running R code or some other software package like that, and just typing values into certain fields. Maybe you had to learn syntax for how to write the code. But oftentimes in these introductory statistics courses, we miss actually understanding what's going on. And I'm going to try to briefly but thoroughly explain statistics to you and how to select the basic statistic tests. So you must understand independent and dependent variables in order to be fluent in statistics at all. But we've talked about it quite a bit. Qualitative and quantitative data. You must understand that. The statistics we talk about will only work with quantitative data. You also need to understand categorical versus continuous data. So this is quantitative data that either falls into categories or along a continuous range. I'll show you an example of that in a minute. And then you need to understand p-values, which I explained twice on the previous slide, but we'll come back to again. Independent and dependent variables. If my previous explanation did not make sense, then please just pause the video here and read this explanation. I wrote this to be, just a little bit different in case it's helpful. Quantitative and qualitative data, same thing. Okay, another slide, same information. I'm not going to mention ordinal data in the lecture here because it's pretty irrelevant to our needs. I can't do that. I will mention ordinal data briefly. Let's say you're taking a survey and people report, let's go with you're doing a survey on quality of life. Well, people who report a 9 or a 10, that's a very big change. To go from a 9 in your quality of life to a 10 in a quality of life where everything's perfect, that's a big jump. But to go from a 3 to a 4, the difference between a 3 and a 4 may not be equal to the difference between a 9 and a 10. Even though numerically the difference is 1, all right, the value of that 1 is not equivalent. Going from 3 to 4, that 1 is maybe less than going from 9 to 10, but they're both 1. That's ordinal data. You'll run into it sometimes. It's usually just mentioned. It's usually not even mentioned in the design, but it's something that's good to be aware of if you're, trying to be a critical consumer of statistics and of literature, okay, of research. Okay, qualitative data, quantitative data, we've talked about it. Very good. Now let's get to the categorical and continuous data. It's really key that you understand this. It's all quantitative, something that can be measured, and two people would measure it the same way, right? We've got categories, our categorical data. Things like high, medium, low, north versus south, improved, unchanged, worsened, all right? Exposed to antibiotic, or antibiotic A, antibiotic B, antibiotic C, right? These are all categories. Some data can fall anywhere within a range of values, like latitude. There's a range of latitudes, but any number within that range is possible. Depth in the ocean or in a lake, any value within that range is possible. Density of bacteria on a plate, density of plants per square meter. Any value within a certain range is possible, like 0 to 100% coverage, things like that. Concentration of lead in water. Any number within a range is possible, okay? That's what we call continuous data. So we have categorical data and we have continuous data, all right? And usually, okay, you will be measuring continuous data. It might be percent transmittance or absorbance. It might be concentration. It might be plant height. All right. We'll usually be measuring things in a with continuous data. Sometimes it'll be categorical. Okay. Sometimes there'll be a treatment and you'll have to say, oh, this improved or it worsened. But that's going to be more. It's going to be a less likely situation. Let's take a look here. P-values, probability that you'd receive the results of your experiment if the null hypothesis is true. Okay? So a p-value of 0.05, it tells you there's a 5% chance that you received the results you did just by chance. Okay? Which could also happen. All right, very good. P-values, where do they come from? So statistical tests, right? They, I'm sorry. One moment, please. We're just going to need to pause this. Okay, our recording should be resumed now. Let's go ahead and get back into it. All right. So, statistical tests in general, they'll put out this p-value. Behind the scenes, there's all this math taking your data, measuring the variance in your data, because you have all these replicates. By the way, you got to have replicates in order to do these statistical tests. Just having one representative from each, from each treatment won't let you run statistics. The test just won't run because they need variance between a bunch of different samples. Okay? They'll do a bunch of equations, produce this value called a test statistic, compare it to some uh, other value that it also generates, and whether that value is higher or lower, it's going to say, hey, good results or bad results, right? I'm hand waving it all because usually what you're mainly concerned with is just the p-value, okay? The p-value gets reported and you want it to be low because that provides more support for your hypothesis. Check this out right here. You're going to encounter 3 types of tests usually. In this class, though, you will also be encountering a chi-squared test. Okay, so we're going to run a chi-squared test in your lab section. Okay, cool. Most of the time, you've got either categorical or continuous numerical independent variables. The explanatory variable, I left the text saying that because it also makes sense with independent variable. Okay, this is the variable you're saying explains the change. The response variable is the dependent variable. I'm using a ball mouse, so it's kind of tricky for me to write. Apologies, but they're dependent versus independent. And you need to know, is your dependent and independent variable either categorical or numerical? Okay? If both your independent and dependent variable is numerical or continuous, okay, I'll put a C-O-N-T right there, continuous, okay? If they're both continuous, then you can run a regression. If the independent variable is categorical and the dependent variable is continuous, then you can run a t-test or an ANOVA. It's all based around how many categories you have for the independent variable. And just don't worry a ton about this. You'll know when to run a chi-squared test. A chi-squared test is run when you have observed values and expected values. And we'll explain that when you hit the lab. Let's just focus today on these three tests, which are the most common for you to encounter, though sometimes you will encounter a slight variation of them. This is another flow chart designed to help you select the right statistical tests. Okay, pretty straightforward. First thing it says is, oh, everything can be measured. Okay, you can actually measure everything. Let's go ahead and look at some examples of what the outputs of these tests or the graphical representations of this data would look like. Here's a t-test on the left. On the t-test, I'm going to Control Z that. I'm going to just put down the laser pointer instead. So in the t-test here, you have a category of before and a category of after. This is your independent variable. Your dependent variable is, it's some percentage, okay? Let's say that it's like percent salinity before and after passing through a certain type of filter, okay? And you're saying, oh, this filter increases salinity, all right? Now you can see from this spread of data here that yes, it looks like sometimes the after group has more salinity than the before group. But is the difference when you average everything significant between the two? For that, you run a t-test. And a t-test that returns a p-value greater than 0.05 indicates that there's no effect of before or after as categories. Over here, we've got an ANOVA on the far right. Same thing. 1, 2, 3 different categorical independent variables. So if there are 3 or more categories, okay, for your independent variable, then you're going to run an ANOVA. Here you've got control, different treatments, and we're saying that they will affect weight of this thing, okay? The control group is important because you want to know what the normal weight of this thing is. All right. What an ANOVA does is it tells you if any of these treatments had an effect, if any of these treatments are different from the other treatments. And if you want to know which treatments are greater than others, well, there's a nice simple follow-up test for that, a separation of means test that you can run. But I'm trying to keep it as simple as possible while still explaining all the statistics that you'll probably need unless you're becoming a statistician. Here is the last group, the regressions, okay? In the regressions you have, let's look at this comparison of height and weight. The hypothesis is that as you increase height, weight increases on average across this population of giraffes, all right? Whatever it is. Those are short giraffes, they're 1.6 meters tall, but you know what I mean. So you have a continuous independent variable. You can measure height of your samples, and it can fall anywhere within a range. And weight can be measured with your samples, and it can fall anywhere within a range. All right? You're saying that there's some correlation between the two. You're proposing that as I increase height, weight will increase. So what you do really is you fit a line to the data, a line that minimizes distance from the line to all the points. And if the line has a slope to it, well, then that indicates a correlation between the two variables. If the line goes up, then that means that as I increase height, I also increase weight. A regression will also spit out an equation for the line, which is really nice. You'll be doing this in lab with standard curves. You're essentially doing a regression, okay? But not to analyze if there is a correlation between two things, rather just to get the equation so that you can make predictions. So regressions have an added benefit of producing a model. Regressions aren't always straight lines. Sometimes there's regressions that go up exponentially. Sometimes there are regressions that fit like sigmoidal lines that are like S-shaped. There's all sorts of different stuff. There's one last thing I want you to look at right here, and that's the R-squared value. While a regression will spit out a p-value for you, the R-squared value is also very important. The R-squared value tells us how well the data fits our line, or in other words, how good the model is as a predictor. All right? In the textbook definition, the R-squared value tells us what proportion of variability in the dependent variable is explained by variability in the independent variable. So an R-squared value of 100% indicates that weight is perfectly predicted by height. And this model, if I put in a value for weight, I can calculate the value for height accurately all the time. All right, pretty cool. Variances, standard deviation, standard errors, null hypotheses, it's all stuff that you can learn more about, talk to me about, ask me about. Variance, though, is very important. You need to see what the spread of data is for a statistical test to work. That's one of the reasons you need replicates. Anyway, we'll come back to these topics on Wednesday of next week, since we have a 3-day weekend. Okay, and we're going to talk about a primary research article very briefly, talk about its different sections, we'll talk about what a meta-analysis is, and then we will get into the week 2 material where we'll really be talking about, where we will really begin discussing all of our chemistry review and the material that you'll be expected to fully understand. Now, as I mentioned before, I want to make it very clear. I'm not planning on testing you in the lecture exam on any of this statistics stuff. Okay? That whole statistics talk was for your own knowledge and edification. It will be useful when we hit a future lab where you have to interpret the output of a chi-square analysis, but it won't be on your test. Anyway, I hope it was informative. I hope you enjoyed yourself watching this lecture. I went over time, my apologies, but thank you for watching, and I'll see you again in person at our next meeting.